Hello everyone. So today we are going to talk about the ketone bodies. We'll talk about the brief introduction of the ketone bodies, and we'll also talk about the ketogenesis, and also we'll talk about how ketone bodies are metabolized in our body. So first of all, you have to know that ketone bodies. There are three ketone bodies, mainly three ketone bodies in our body. They are acetone, acetoacetate, and beta hydroxybutyrate. So mainly in the liver, the amino acid is converted into the acetoacetate and beta hydroxybutyrate, whereas the acetone is the spontaneous occurrence of the acetoacetate, and it is eventually expired by the lungs. So what happens in the ketone body is that in prolonged starving and diabetic ketoacidosis, the oxaloacetate is depleted for the gluconeogenesis. So in alcohol excess, so if there is excess consumption of the alcohol, though this sends oxaloacetate to malate, which build up the acetyl coenzyme, and it is shunted into the ketones body synthesis. So this also explain the hypoglycemia in the people with the prolonged alcoholism, or the acute alcoholic intoxication. So why there is hypoglycemia in the alcohol intoxication, this also explain this. There will be the oxaloacetate will be depleted for the gluconeogenesis. So when there is alcoholism, due to the excess alcohol consumption, there will be the excess NADH will be shunted to oxaloacetate to malate. So it will build up the acetyl coenzyme, and this acetyl coenzyme, instead going to the TCA cycle, it will be shunted into ketone body synthesis. So what will be the clinical features of the excess amount of ketones in our body? So the breath smells like acetone. So acetoacetate will be eventually converted into acetone, and it will be expired by the lungs. So the smell of the acetone is like fruity smell, and from the urine the acetoacetate is excreted out. You have to know that the RBC can't utilize the ketones. The other bodies like extraskeletal or extra hepatic tissue will utilize the ketones, but RBC can't utilize the ketones. And you have to know that the HMG CoA lyase is required for the ketogenesis. Now let us discuss the ketogenesis. So you know that for the formation of the ketone bodies, the fatty acid and amino acids are required. So the fatty acid and amino acid will be converted into acetyl coenzyme, and acetyl coenzyme, upon the action of beta ketothiolase, it is converted into acetoacyl coenzyme. The acetoacyl coenzyme, with the help of HMG CoA synthetase, and with the action of the acetyl CoA, it is converted into HMG CoA. So HMG coenzyme A synthetase is the rate limiting enzyme. So after the formation of the HMG coenzyme, with the action of the HMG coenzyme lyase, it is converted into the acetoacetate. So this acetoacetate is the one of the ketone bodies, and you have to know that this acetoacetate will be transported to the circulation, and the carbon dioxide will be excreted out. That means one carbon atom will be taken out from the acetoacetate, and this acetone is formed, and this acetone is expired by the lungs. In acetoacetate, upon the action of the beta hydroxybutyrate dehydrogenase, it is converted into beta hydroxybutyrate. So this beta hydroxybutyrate will go to extrahepatic tissue, and also the acetoacetate will also be utilized for the further metabolism. That means the beta hydroxybutyrate and acetoacetate both are interchangeable. In this, both beta hydroxybutyrate and acetoacetate can be used in the extrahepatic tissue. How it is used in the extrahepatic tissue? This beta hydroxybutyrate or this acetoacetate will be transported to the TCA cycle, and this from this TCA cycle, energy can be generated. Also, from these two molecules like acetoacetate and beta hydroxybutyrate, with the help of TCA cycle, it is converted into acetoacyl coenzyme, and this acetoacyl coenzyme will be eventually converted into acetyl coenzyme, and this acetyl coenzyme can again be used in the TCA cycle for the generation of the ATP. But you have to know that the ketone bodies can only be utilized by the extrahepatic tissue. The RBC cannot utilize the ketone body. So in the event of the diabetic ketoacidosis, or in the event of the prolonged starvation, the RBC can't synthesize the enough ATP for its energy if there is excessive ketogenesis. Only extrahepatic tissue can utilize the ketone bodies. So this is the main thing you have to know about the ketone bodies. So today we have discussed about the types of ketone bodies, what happened in the prolonged starvation and diabetic ketoacidosis, why in the alcoholism there will be the hypoglycemia due to the shunting of the oxaloacetate to malate, and we also discuss about the various clinical features of the excessive ketone bodies in our body. We discuss about the ketogenesis, and we discuss about the metabolism of the ketone bodies in our body, how the ketone bodies are utilized in our body. I hope you understood this lecture. Thank you.